Hello, students. This time I am going to give you some concept how to screen the transformed colony, which is a very important step in order to confirm whether the plasmid has or any vector has integrated DNA or not. Basically, this is a very tough task and lot of concepts are required to understand the screening process. Here I am taking three vectors and these vectors are PBR 322, Buck 19. And with these two vectors, the DNA is integrated. And let us just talk about when the DNA is integrated, how it can be screened. So, when a vector gets any DNA, then this is called recombinant DNA, chimeric DNA, because it has got the two parts. One belongs to the plasmid vector, and another one is the foreign DNA. So for the cloning process, we must be ensured whether the DNA has been integrated or not. So once the DNA gets integrated, the bacteria which harbors this integrated DNA with the vectors, they are said to be as transformants, and the process is transformations. So it is an old concept that was developed in 1928 by Frederick Griffith, where he was finding whether the what is the genetic materials, whether it is the DNA or the proteins. And during his experiment, he found that when a rough strain of the bacteria, that is Pneumococcus bacteria, is mixed with the heat killed smooth virulent bacteria, then that mouse tends to die. So let us just first understand what is the Griffith experiment of transformation. So pneumococcus bacteria that exist in two different forms. One is having the rough surface, another one is having the smooth surface. So what happens when this rough surface is inoculated inside the mouse? Then within the mouse, the antibodies, they tends to bind with the rough surface. They tends to bind with the rough surface, and that leads to the neutralization of these very bacterial strain. So there is no any bacteria, and so the mouse becomes the healthy. So it is called as the non-virulent. And when we take the smooth one, so a smooth one is unable to bind with the antibodies. They are unable to bind with the antibodies, and so this bacterial strain remains there. It grows and it kills the mouse. In another experiment, this bacteria, a smooth one, that is heat killed, it is degenerated. So there is no bacterial colony grows inside the mouse, and mouse will be healthy. But what happens in this case when the this is the live one and this is the dead one? So when this dead one containing the DNA get integrated into the live one, then some of the live they get transformed into the smooth one, and the culture of that smooth that leads to the death of the mouse. So this bacterial stains, this is the R1, they get transformed into the smooth one. How? Because in this, the DNA of this this they get some of the DNA of the smooth, and due to this integration, the R get transformed into the smooth one, leading to kill. So that process is called as transformation. So, and it was further proved that it is the one of the DNA which is responsible for the transformation of the R strain into the smooth strain. So, based on this transformation experiment, the screening for recombinant DNA was conceptualized. So, recombinant DNA, you better know that it is a technology to produce DNA with integrated heterologous DNA, that DNA which belongs to the other organisms. And the RDT or the recombinant DNA technology that involves the two process, that is the cloning and the expansion. So this recombinant DNA or the chimeric DNA, it is needed for the cloning the or making the more copies of the DNA, as well as allowing it to express into the proteins. So here you can see this is the vector PBR322, which is having the TET-R regions, then the ampicillin resistant regions, then the oride for the replications, and many of the some of the restriction sites are there. So when there is no any DNA is inserted into these regions or in these regions, then this is called as the non-recombinant. And when this plasmid, when it will introduce into the bacteria, this is the bacterial genome, and this get inside this one, and if there is no any integrations at all, then this is called as the non-recombinant plasmid, and the bacteria is called as non-transformed. But in this case, when the DNA, this heterologous DNA, they get inserted into the BAM H1 regions, okay, and BAM H1 region is present within the tetracycline resistant regions. So when it will be integrated, then this TET-R will be transformed into the TET-S, because the reading frame of this TET region is disturbed. So such a plasmid is called as recombinant plasmid, because it has got the plasmid DNA as well as the this heterologous DNA. Okay, so this is a plasmid, and when this will be introduced into the bacteria, so it will be having the uh, this regions. This is the integrated DNA, or you can say with the color also. So let us this is the color. So this is the integrated DNA. So this bacteria is called as the transformed bacteria. So there is a difference between the non-transformed and the transformed bacteria, and better be called as the transformants and the non-transformants. Integrated DNA, when it is present in the plasmid vector, it is called as the transformants. When not present, it is called as the non-transformants. So how to screen? So though screening is a process to just differentiate whether the DNA has integrated or not. So we can just conduct one experiment, and the experiment is that you will have to just make a colony to grow a colony in the culture medium. So what you will do, you just prepare a culture media plate, and culture media plate, you have got the samples of the inoculum. So you just pour this inoculum into the culture media, and keep it at 37 degrees centigrade in the incubator, and you after 24 hours you can get the result, that colony will grow. So, and for the introduction into the host cells, we require uh, uh, we require uh, we require the calcium chloride and the high temperature, because that will allow the introductions of the plasmid vector containing the DNA into the host cells. So, what things you can do? You just grow the colony, transformed colony. Okay, you are having the two colonies. That is one is transformed and another one is not transformed. Okay, you better know that the transformed will be having the DNA, and non-transformed will be like this. So Uh, so when you grow this colony into the culture medium, so both of the colony will grow because it is also having the AMP, which is not disturbed, and it is also having the AMP. So this is not disturbed, both the colonies. So both the colonies will grow. ठीक है, transformed will be also grow and non-transformed will grow. But again, here is the difficulty. What is the difficulty? That how you can differentiate which one is transformed, which one is not transformed. So what will you can do? You just make a replica. So how you can make a replica? You just take a one that is called as the stamp, and this stamp is covered with the velvet paper, and that should be sterilized. So you keep this velvet paper and make a print. Okay, and now in another culture media plate, which is having the tetracycline. Okay, which is having the tetracycline. Okay, no AMP, only tetracycline will be there. So what will happen? So this the those bacteria which is not transformed, yeah, it means that not transformed means what? The AMP R will be there and the tet R will be there, non transformed. So they will grow. So those bacteria which are growing, they are called as the non transformative. And the bacteria which is not growing, this will be the AMPR and the TET-S. And due to presence of this tetracycline resistant, since bacteria has lost the resistant, therefore it is killed. So you can just take the positions. What is the positions of that? So this is the one position. Of course, this is the one position. This is the two position. This is the two position. This is the three position. This is the three positions. This is the fourth position. This is the fourth position. This is the fourth position. This is the fifth one. So this is the you can say this is the fifth one. This is sixth one. This is also sixth one. This is seventh one. This is seventh one. This is the seventh one. This is eighth. This is eighth. This is ninth. This is ninth. This is tenth. This is tenth, and this is 11th. This is 11th. So comparing these two, what you will find that this number 3 colony is transformant. This number 7th colony, it is transformant, and this number 9th colony, it is transformant. So out of this, you can just compare and you select this colony, you select this colony, you select this colony, and this colony will belong to this very form, that is having the integrated DNA. So it is very easy to just make a media plate. You pour the required tetracycline or the ampicillin and you select them, but you will have to make a copy of this one. And how to make a copy of this colony? You will have to use a stamp with the velvet paper, and that should be sterilized, and you just make a print over the this one, another media plate. So positions you can know. What could be the positions? Another vector is the PUC 19, or you can say the PUC 19. So this was generated in order to just get rid of the some of the limitations which we get in the PBR 322, because that was a very lengthy process. You will have to culture in the ampicillin or the tetracycline. So in order to avoid this complexity, the another vector was created, or it was genetically engineered. And this PUC19, it possesses a one gene that is called the Lacjet genes, which was integrated, which was attached within the multiple cloning sites and real reading frame. And in order to express, in order to allow its expressions, the or regulatory genes LAC-I was there. So LAC-I, that basically it synthesizes a repression molecules, and LAC-Z, it synthesizes the beta galactosidase. So beta galacto beta galactosidase enzymes. So what beta galactosidase enzymes does, it just break the lactose, which is a disaccharide, into the two molecules of the monosaccharide, that is the glucose and the galactose. So and this undergo during the pathway of the metabolism to produce the energies. And you also know that this lac I is inactivated in the presence of the invisible molecules, which is lactose or allolactose. So let us just see that how the concept of the lac operon can be used in order to screen out the transformants and the non-transformants. So before that, we must understand what is the lac apron. So lac apron is a cluster of genes which includes the this is the lac I genes and the three structural genes Z, Y, A. Okay. So here I am only dealing with the lac Z genes. So what happens? This lac operon is has got the two mode. Either it will be a switch off or it will be switch on. Switch off or it will switch on. Okay. So when there is no lactose present, no lactose present, that is no lactose, that is no lactose absent. So what will happen? No lactose present, so then this repressor will form because it has got the promoter, its own promoter. So this repressor will tends to bind with the this operator regions. It will bind, and when the operator is bind, then there is no synthesis of this one. So repressor binds to the operator regions, preventing the RNA polymerase from the transcribing the operon. So no galactosidase. And in such condition, it will be the switch off, and there will be no growth of the colony because there is no energy. Okay, no growth of the colony will be there. But when what happens when the lactose is present? When the lactose is present, then it will inactivate the repression molecules. It inactivate the repression molecules, and when it will inactivate, the repressor is unable to bind. There is no binding at all to the operator. So this is open. This operator is open, and when this operator is open, the RNA polymerase, RNA pol, it can move and it will synthesize the lac messenger RNA, resulting into the expression of the beta galactosidase, permease, and the transacetylase. And when such thing will happen, the bacteria will tends to grow. So there is a bacteria will. So it means that there is no colony. There is no colony uh, in the absence of lactose, and in the presence of lactose there will be the colony formations. So utilizing this very concept, the screening was done for the PUC19 with the integrated DNA. So this is the structure of the PUC19, which possesses the some of the 268, like more than 2,000 base pair, but less than 3,000. Uh, and it has got the LAC-Z genes. The LAC-Z gene, it has been just integrated at the 5' end of the multiple cloning sites. And the LAC-I gene, because it is in toward the upward and it is toward the downward stream and the upstream. So it is in the downstream and it is in the upstream. So when there is no integrations at all, the plasmid or the Lacjet is intact. So not transformed with the intact Lacjet. And this is the transformed when the this clone DNA gets integrated into this one. So basically the main major concept is that the transformed, it will be having the extra DNA in this regions, and it won't have the any DNA molecules. So here we are using the IPTZ, isopropyl beta d-thiogalactopyranoside, and basically it is a inducer molecules. So you can say that this is lactose and IPTZ. These are these two are the inducer, and but structure different, therefore it is called as the analog. So it is the analog of the lactose, which means that structure is different and functions are same. So in presence of IPTZ, in presence of IPTZ repressors product, in presence of the IPTZ repressor product of lac I is inactivated. Lac I is inactivated. It means that when the IPTG is present, IPTZ is present, IPT is present, then the repressor will binds with the IPTZ. When this what do you call, yes, IPTZ is present, IPTZ is present, then the lac I, which will produce the repressor, which will lac I which will produce repressor. It can bind with the IPTZ. It will bind with the IPTZ, and it will inactivate the repressor molecules. So when it will be activated, the product will formed. It means that the Lacjet will express. Though enzyme production is induced, okay, and galactosidase is formed. But when this is this because this insertion was taken place within the reading frame, so this reading frame Lacjet is disturbed, so there will be no lac expressions. Even in the presence of IPTG dead, the repressor is inactivated. But since the Lacjet gene is disturbed, Lacjet gene is inactivated. It is also inactivated due to insertion. So there is no expression, and the galactosidase is not formed. So you can understand how this can happen. So in the presence of the IPTZ, there is the expression, but in the presence of IPTZ, there is no expression because the Lacjet gene gets disturbed. So how you can screen out, how you can visualize these transformants. So these transformants can be visualized by using a indicator, and that indicator is the XGal. So XGal is the substrate for the IPTZ. XGal is the substrate, that is the 5-bromo 4-chloro 3-indole beta D galactopyranoside. This is the XGal. So when this beta galactosidase, this Lacjet is expressions. Okay, there is the IPTZ. Okay, you just write IPTZ present. So repressor is inactivated, and this expression will goes on. The beta galactosidase will form, and this beta galactosidase, it will do, it will just interact with this one. It will interact with this one, leading to the dimerizations, and it forms a blue precipitate. Okay, so blue precipitate will form here. uh, you can say this is the blue precipitate will form. But if the gene gets integrated, gene gets integrated. That's the foreign DNA. Foreign DNA. This is the foreign DNA within the Lacjet regions. So what will happen? This XGal will be not binds with the enzyme because enzyme is not present. Basically, it binds with the enzymes and the product gets dimerized to form the blue product. But here, the no enzymes are present. So these reactions won't happen. These reactions won't happen, and only the X-gal will remain as such. When X-gal will remain as such, then what will happen? Then in that cases, the there is there that this will remain as such, and only the white white precipitate, or you can say the white color will be there. So when there is no insertions, the blue will form. When there is insertions, then the white will form. And this reaction was taken as a background for screening. So you can see here, this is the when the no lac jet was expressions, no Lacjet expressions, it means that the reading frame of the Lacjet is disturbed. In the presence of induced hypertrophy, X-gal remain as such, leading to the formation of the blue white colony. So here you can say this colony is white, this colony is white, this colony are white. It means that they are transformants. And here the blue colony, in this blue colony, what happens? The X-gal reacts with the enzymes, that is galactosidase, leading to the dimerizations, and the blue precipitate is forming. And here there is a X-gal remain as such, that is minus enzymes. This is the white. So no enzymes are there when the things are getting integrated. Another thing that another it was nowadays it is very much used, the use of suicidal genes in the bioremediation process. So here what happens, the in the correct reading frame, in the correct reading frame of the Lacjet, it is the Lacjet, in the correct reading frame of the Lacjet, another gene, suicidal gene, was integrated. So this suicidal genes was integrated in the plasmid vector. So this plasmid vector, what happens in the PUC19? This is the PUC19. So what happens? There is the AMP and there is the LACZ. Okay, LACZ, and also the PI is there. So just like the lac operon, just like the lac operon, here the LACZ is gene is there. Here the ZYA. So instead of this Y, it has got the suicidal genes. So once this is operated, then it will also synthesize this and it will also synthesize this proteins. So Lacjet beta galactosidase will form, and also the suicidal genes product will be also formed. So here some of the concepts are there. What happens? This is the DNA. Okay, this is the you can say the plasmid DNA, plasmid DNA or any DNA. Okay, so when the suicidal gene is product is present, when the suicidal gene product is present or it is synthesized, then what happens in the usual process when a DNA breaks, it forms the breakage in the DNA. But due to presence of this suicidal genes, it will prevent the DNA repair mechanisms. So there will be no DNA repair process in the presence of suicidal genes, and the for example this can be sealed by means of the ligase. Okay, ligase. So this product that hamper the ligase enzymes. So what will happen? This DNA gets accumulated inside the cells, and when the DNA gets accumulated inside the cells, it has got the harmful effect, and there will be bacteria is killed. Bacteria is killed because there is no any intact DNA to promote the growth. So bacteria is killed in the presence of the suicidal gene expressions. But if the suicidal gene expression is lost, no suicidal gene expression is there, then what happen in the usual process? The DNA will be breakage to form this one, but here the ligase activity is not hampered, it is not prevented, and so this ligase will seal the DNA and intact DNA will form, and this intact DNA will promote the growth, and that's why bacteria are not killed and the colony will formed in the culture medium. So this concept was taken as a considerations to screen out the transformants and the non-transformants. So here you can see here, when the culture media containing no IPTZ, when the culture media, when the culture media doesn't contains any IPTZ, that is inducers are not there, and the plasmid is also intact. Therefore, there is no killing, no killing. You can see here the lac I, it is activated. Okay, it is forming the repression. Okay, lac I, lac I is expressed. Okay, like it will form the repression. Okay, in the presence of no IPT, because it has got its own promoter. So in the presence of no IPTZ, this lac I is produced, and this lac I, it will bind with the operator regions. When it will operator regions is, when it will bind, so there is no expression of the lacjet also no expression of the suicidal genes. So due to absence of this lacjet, there will be no growth, there will be no growth, and suicidal gene is not produced, it doesn't have any effect. So it is due to the absence of lactate, that is no colony is formed. So you just better stand in the when this one, this is due to the switch off, it is the switch off condition. As usual, that is IPTZ is not present, but when IPTZ is present, then it will be switch on, switch on. So what will happen here? The this lac z will express, which will induce the growth, and the suicidal gene is inactive. It means that the bacteria will not killed. So when the bacteria will not kill, then there will be the growth. So all this bacteria will growth because Lacjet will promote the growth, and suicidal gene is no suicidal gene is there, and therefore non-functional suicidal protein leads to the leads to the you can say no killing of the colonies. So in the this colony, and this is the case when there is a transformation, that is the integration. It means that the suicidal wild type is converted into the suicidal mutant type. Even the IPT is present. So IPT is inducers. Inducer means it will just inactivate the repressors. So this is the inactive, lac I product is inactivated. It means that repressor is inactivated. The repressor is activated, repressor is sorry, this is the repressor is activated in the absence of the IPTG, and the repressor is inactivated in the absence, in the presence of the IPTG. So no IPTG is there, it means that repressor is activated, it will bind, and if repressor is inactivated, it won't bind to the operator, it won't bind to the operator. uh, so uh, in third case, when the no insert is there, here insert is there, IPT is there, it means that it is also the switch on condition, it is also switch on. But here you can see, here you can see, here you can see that no insert is there within the Lacjet. Okay, no insertion is there, Lacjet. So what will happen? No insert and IPTG will induce. It means that the repressor will be lac I product is inactivated. Okay, and it will be switch on. Okay, but since the Lacjet is present, it will promote the growth, but same time the suicidal gene is also there, so whatever the growth will be there, they will be killed. So due to presence of these two together, they will not produce any colony, so there will be no colony. So what you will see here, you can have this two experiments. When the colony will be formed, then there is integration, it will be there. Yani this represent the transformations. And when there is no integration, no colony will be formed. So you can have the screening by these two methods. When the colony will be formed, it is the transformations, and when no colony will be formed, transmission. And but the condition is that IPT is present and IPT is present, but insert is taken place in the transformation, no inserts taken place in the no insert, that is non-transformations. So this is the simple concept how to screen out the transformants and the non-transform during the recombinant DNA technology process. I think this is the simplest way. In case if you feel any queries, you are invited to ask any questions, and I will try to explain these things. So with this little informations, I must say thanks for watching and keep in touch. Thank you very much.